The title of our paper is Nanopore Ion Sources Deliver Ions of Individual Amino Acids and Peptides Directly into High Vacuum. It appears in Nature Communications. Mass spectrometry is the workhorse of proteomics research. The technique accurately determines the mass-to-charge ratios of vapor phase ions. For proteomics, it's first necessary to turn the interesting proteins, peptides, and amino acids that are natively found in solution into vapor phase ions. The main technique for doing this is called electrospray ionization, or ESI for short. It works as illustrated here. We squeeze the liquid sample through a small tube while applying a voltage to the liquid. Charged droplets are released into a background gas that helps the solvent evaporate. The drops undergo a string of Coulomb explosions that spread them out into a wide plume. Lots of analyte is lost in this process because the droplets fly in all directions and most of them crash into various parts of the instrument. Only about 1% of the analyte actually makes it into the mass spectrometer. This low transfer efficiency is a fundamental problem, especially when one wants to measure small samples like single cells or single molecules. But there is hope. The very last phase of electrospray ionization doesn't occur until the charged droplets shrink down to just a few nanometers in size. At that scale, Coulomb forces drive ions directly out of the liquid and into the vacuum by a process called ion evaporation. These are the vapor phase ions that a mass spectrometer ultimately measures, and this gives us an idea.
So the big idea is to shrink the ion source down to just tens of nanometers across, the size of those final ESI droplets, and stick it directly into the high vacuum region of the mass spectrometer. That way, we should be able to emit ions directly by ion evaporation and skip the lossy Coulomb explosion cycle, which is inherent to ESI. These are very simple to fabricate out of quartz. With the right recipe, you can easily pull tips down to around 20 nanometers using a standard laser-based pipette puller. In order to test the ion source, we built a custom mass spectrometer that can house the ion source inside a vacuum chamber, along with ion optics, a quadrupole mass filter, and a single ion detector. The pressure inside the chamber is low enough that we don't expect an ion to bump into even a single gas molecule en route to the detector, located almost a meter away. The Nanopore tip is easily attached to a custom kinetic mount that supplies voltage and fresh sample solution to the tip while holding vacuum. We use an identical mounting system outside of the vacuum chamber, which is equipped with a microscope to ensure that the ion source is properly aligned. This is an example of a typical amino acid mass spectrum generated using a Nanopore ion source and our home-built instrument. Here we used an aqueous solution of arginine with some acetic acid to reduce the pH and protonate the arginine molecules, giving them a positive charge. In the spectrum, we see a peak that corresponds to bare arginine ions, and we also see a series of peaks that corresponds to arginine ions with a small number of water molecules attached. So sometimes we pull out bare ions, and sometimes we pull out hydrated ions. We also see some peaks that correspond to small protonated water clusters at a lower mass to charge ratio. We've measured spectra of 16 different amino acids using the Nanopore ion source, all detected primarily in their unhydrated states. In several spectra, we also see the first or second hydrated states and protonated water clusters at low mass. We are still working to understand the variance in the distributions of solvation states, but the possibility of generating spectra containing just a single unhydrated peak is encouraging as it makes interpretation of the spectra very simple. The Nanopore ion source also emits small peptides. Here we see a mass spectrum obtained from a mixture of glutathione, a 3 amino acid long peptide, and angiotensin II, an 8 amino acid long peptide. The angiotensin ions appear in the +2 and +3 charge states. So the Nanopore ion source can generate many of the same kinds of small analyte ions as conventional ESI sources. We also use the Nanopore ion source to measure the spectra of 2 post-translationally modified variants of glutathione and show that these PTMs are easily distinguished. Recall that a critical bottleneck in ESI is the ion transmission efficiency of the ion source. Typically, only around 1 in 100 ions makes it from the high pressure region of the ion source into the low pressure region of the mass spectrometer. We set up a simple experiment demonstrating that the Nanopore ion source circumvents this bottleneck by delivering ions directly into high vacuum. We measured the current leaving the ion source, as well as the current striking a downstream Faraday cup, and we find that over 90% of the ions are transmitted from the source to the detector. This is approximately 2 orders of magnitude higher than the typical ion transmission efficiency of nano-ESI. Now, a reasonable question to ask about these results is, how do we know that the current in these measurements is actually composed of ions rather than charged droplets? So we added a magnetic sector to the instrument to separate charged nanodroplets from small ions, and we find that indeed, the current is composed entirely of small ions. No charged droplets are measured. The ion evaporation mechanism, in which ions are pulled directly out of the liquid by strong electric fields at the surface, requires that the analyte be charged in the liquid in order to be emitted. pH controls the charge state of amino acids in solution, so we expect the amino acid ion signal to depend strongly on the pH. Here we measured the mass spectrum for a mixture of arginine and sodium iodide, where the sodium ions serve as a reference ion, which is independent of pH. At high pH, where most of the arginine is neutral, the arginine signal is very weak relative to sodium. When the pH is reduced, positively charging the arginine, we see a strong arginine signal appear relative to sodium, just as the ion evaporation mechanism predicts. This contrasts with findings from conventional ESI, where the ionization efficiency of amino acids is found to have a very weak dependence on pH. All the evidence we've gathered indicates that the nanopore ion source emits ions directly into vacuum by ion evaporation rather than the conventional electrospray process. This is fundamentally due to the size of the nanopore tip, which enhances electric fields and increases the Rayleigh limit needed to break surface tension and emit charged droplets. For sub-100 nanometer tips, ion evaporation is energetically favored over charged droplet emission.
In conclusion, we've developed and characterized a nanopore source that delivers ions of amino acids and peptides directly into high vacuum. The source produces ions by the mechanism of ion evaporation rather than the conventional electrospray mechanism. This new emission mechanism, which involves strong electric fields essentially pulling ions directly out of the liquid into the vacuum, and the absence of a background gas when it gets there, enables the ion transmission efficiency to approach unity. Looking ahead, we plan to develop a nanopore ion source to measure individual proteins. We want to fragment a protein and identify the pieces, ideally in a way that provides information about their original arrangement in the protein. We dream of ultimately sequencing a single protein by breaking it into its constituent amino acids and identifying them in order by mass spectrometry.